epigenetic control of gene expression. So the definition of epigenetics is heritable changes in gene function without changes to the base sequence of DNA. So in eukaryotes, epigenetic control can determine whether a gene is switched on or off. Therefore, whether the gene is expressed, transcribed and translated or not. It works through the attachment or removal of chemical groups known as epigenetic marks to or from DNA or histone proteins. These epigenetic marks don't alter the base sequence of DNA. They alter how easy it is for enzymes needed for transcription to interact with and therefore transcribe the DNA. Epigenetic changes to gene expression play a role in normal cellular processes and can also happen in response to changes in the environment. For example, pollution and food availability. Inheriting epigenetic changes. Organisms inherit DNA from their parents. Most epigenetic marks on the DNA are removed between generations, but some aren't removed and are passed on to offspring. This means that the expression of some genes in offspring can be affected by environmental changes that affected their parents or grandparents. As an example, epigenetic changes in some plants in response to drought have been shown to have been passed on. So there are several epigenetic mechanisms used to control gene expression. The first is methylation of DNA. The second type you need to know is acetylation of histones. So methylation is where a methyl group, which is a type of epigenetic mark, is attached to the DNA coding for a gene. And the group always attaches at a CpG site. This is where a cytosine and a guanine base are next to each other, linked by a phosphodiester bond. And if there is increased methylation, this changes the DNA structure so that the transcriptional machinery, like the enzymes, can't work with the gene, and therefore the gene is not expressed. The second type is acetylation of histones. So histones are proteins that DNA wraps around to form chromatin, which then makes up chromosomes. Chromatin can be very condensed or less condensed, and how condensed it is affects the accessibility of DNA and whether it can be transcribed or not. Histones can be epigenetically modified by the addition or removal of acetyl groups, which is another type of epigenetic mark. When histones are acetylated, the chromatin is less condensed. This means that the transcriptional machinery can access the DNA quite easily, and therefore the genes are transcribed. However, when the acetyl groups are detached, known as deacetylation, from the histones, the chromatin becomes really highly condensed, and therefore the genes in the DNA can't be transcribed because the enzymes involved in transcription can't actually access the DNA because it's so tightly wound. And histone deacetylase, or HDAC enzymes, they are responsible for this removal of the acetyl groups, which then makes it really condensed, and therefore you can't transcribe the DNA. So epigenetics can play a role in the development of some types of disease. For example, abnormal methylation of tumour suppressor genes and oncogenes can cause cancer. And it can also play a role in the development of many other diseases, including Fragile X syndrome, Angelman syndrome, and Prader-Willi syndrome. So Fragile X syndrome is a genetic disorder. The symptoms are learning and behavioural difficulties and characteristic physical features, like you can see here they've got large ears, prominent forehead, etc. And it's caused by a heritable duplication mutation in a gene on the X chromosome called FMR1, and the mutation results in the short DNA sequence CGG being repeated more times than usual. These repeats mean that there are more CPG sites in the gene than usual, and more CPG sites results in increased methylation of the gene, which switches it off. Because the gene is switched off, the protein is not produced, and this lack of protein is what causes the symptoms. Angelman syndrome is a genetic disorder which affects the nervous system. The symptoms are delayed development and motor problems. It's caused by a mutation or deletion of a region of chromosome 15, usually on the maternal allele. In the affected region of chromosome 15 is missing, and the paternal allele is present in the cell, but it is switched off by methylation. So the gene is not transcribed, meaning a protein is not produced, which leads to the symptoms of the disorder. Prader-Willi syndrome is a genetic disorder characterised by developmental issues and excessive hunger. It's caused by the loss of function of genes from a region of chromosome 15. It is the paternal allele that's usually transcribed, and so the syndrome results in when the deletion occurs on the paternal chromosome. The gene is silenced by methylation and so is unable to compensate, and the lack of the protein encoded by this gene leads to the disorder. Treating disease. So epigenetic changes are reversible, which makes them good targets for drugs to combat the diseases they cause. These drugs counteract the epigenetic changes that cause the disease. For example, increased methylation is an epigenetic change that can lead to a gene being switched off. Drugs that stop DNA methylation can sometimes be used to treat these diseases. The drug acetylation is used in chemotherapy for types of cancer caused by increased methylation of the tumour suppressor genes. Tumour suppressor genes usually slow cell division, so if they're switched off by methylation, the cells are able to divide uncontrollably and can form a tumour. Acetylation inhibits the methylation of these genes by blocking these enzymes. Decreased acetylation of histones can also lead to genes being switched off. So HDAC inhibitor drugs like romidepsin can be used to treat these diseases, including subtypes of cancer. They work by inhibiting the activity of histone deacetylase or HDAC enzymes, which are responsible for removing the acetyl group from the histones. Without the activity of HDAC enzymes, the genes remain acetylated and the proteins they code for can be transcribed. The problem with developing drugs to counteract epigenetic changes is these changes take place normally in a lot of cells, so it is important to make sure the drugs are as specific as possible. For example, drugs used in cancer therapies can be designed to only target dividing cells to avoid damaging normal body cells.